Today we will continue our discussion on the different cell types, their parts, and corresponding functions. As mentioned in our previous lecture video, prokaryotic cells are considered simpler compared to eukaryotic cells. Eukaryotic cells have membrane-bound organelles not found in prokaryotic cells. Plant, animal, protist, and fungi are composed of eukaryotic cells. Eukaryotic cells contain highly organized structures with organelles and structures found in specific location in order to function efficiently. The plant and animal cells are generally used to understand the different structures of a eukaryotic cell. Just looking at the figure, what is the immediate difference between an animal and plant cell? You're right, it's their shape. Animal cells' shape and sizes vary greatly from irregular shapes to round shapes, most defined by the function they perform. Plant cells are larger than animal cells, with the cell size ranging from 10 micrometer to 100 micrometers in length, and plant cells are also similar in shape, with most cells being rectangular or cube-shaped. The different cell parts will be discussed based in order of their functions. First, hereditary information with the nucleus and ribosomes. Endomembrane system with endoplasmic reticulum, Golgi apparatus and vesicles. Cellular digestion with lysosomes and peroxisomes. Energy converters with chloroplast and mitochondria. Structural support with the cytoplasm, plasma membrane, cell wall, and cytoskeleton filaments. Cell communication with plasmodesmata and junctions. Movement, centrosome and centrioles, and cilia and flagella. And finally, for storage, vacuoles. Okay, the first parts are those involved in hereditary information. Do you know what directs the activities within the cell? The nucleus, or the brain or command center of the cell, contains almost all of the cell's hereditary information, or DNA. It is spherical or oval in shape and the largest structure in the cell. It is enclosed in a nuclear envelope, a double membrane with a nuclear lamina to provide structure and shape. Found along the envelope are nuclear pores that act as channels, allowing exchange of materials between the nucleus and cytoplasm, and spherical bodies known as nucleoli can be found inside the nucleus and are involved in rRNA synthesis, which is necessary for the production of ribosomes. Ribosomes are the site for protein synthesis. A ribosome is functionally active when two of its subunits are joined together and are made up of RNA and proteins. There are two types of ribosomes, free and membrane-bound. The free ribosomes, found in both prokaryotic and eukaryotic cells, synthesize proteins to be used inside the cell. Membrane-bound ribosomes, only found in eukaryotes and located in the endoplasmic reticulum, synthesize proteins destined for insertion in the plasma membrane or export from the cell. The endomembrane system is a group of membrane-bound organelles involved in the transport of materials, specifically proteins, into and out of the cells. Proteins synthesized by ribosomes enter the endoplasmic reticulum. The endoplasmic reticulum is composed of an extensive network of flattened membranous sacs or tubules called cisterns. It serves as a connection between the nuclear envelope and the Golgi complex. The endoplasmic reticulum, continuous with the nuclear envelope, contains ribosomes. This part is called rough endoplasmic reticulum or RER. The RER appears to have bumps on it. Its interior consists of sac-like structures called lumen where folding and processing of proteins occur. The smooth endoplasmic reticulum or SER with few or lacking ribosomes extends from RER to form a network of membrane tubules. The SER contains enzymes to synthesize phospholipids, fats, and steroids. It is also involved in detoxification of drugs, releasing glucose, and contraction, triggering the release of calcium ions. Proteins exit the endoplasmic reticulum and are transported to the Golgi apparatus by means of transport vesicles. The Golgi apparatus is also known as the Golgi complex or bodies. It consists of flattened membranous sacs called cisternae. It is considered as the post office of the cell. Why post office? Because proteins are received, processed, sorted, and packed in the Golgi apparatus. The proteins enter the cis phase of the Golgi apparatus. Proteins exit the trans phase of the Golgi apparatus and move to the plasma membrane by means of secretory vesicles. Proteins are then secreted from the cell. Vesicles are inclusions with lipid bilayer membranes that aid in cellular transport inside and outside the cell. There are two types of vesicles mentioned earlier. The transport vesicles move molecules within the cells, while the secretory vesicles excrete molecules from the cells. The next parts are involved in cellular digestion. Molecules that enter the plasma membrane and some secreted within the cell need to be digested or broken down first before the cells can use them. Lysosomes, the digestive plant of an animal cell, are formed from the Golgi apparatus. It is spherical, composed of a single membrane, and lacks internal structures. It contains powerful enzymes that are capable of breaking down various molecules and harmful bacteria that enter the cell. Molecules are delivered to the lysosome for digestion by means of vesicles. Lysosomes are also known as suicide bags since it can digest damaged organelles, and the release of its enzymes into the cell can cause cell death or apoptosis. White blood cells contain large numbers of lysosomes to break down harmful bacteria entering the body. Initial reports on the existence of lysosomes in plant cells did not find general acceptance. Instead, plant vacuoles that are structurally and functionally similar with animal lysosomes established that vacuoles represented lysosomes in plant cells. Peroxisome, similar in structure to lysosome but smaller, is the site for oxidation reactions for both plant and animal cells. Peroxisome contains enzymes that can break down various organic molecules and toxic substances through oxidation. In the process of digestion, hydrogen peroxide, a toxic substance, is produced. This byproduct is further degraded within the peroxisome to water and oxygen. Aside from peroxisome, plant leaves have glyoxysome, which are involved in the breakdown of fatty acids. The next two organelles are energy converters. Most of the activities within the cell involve the production and consumption of energy. Two major organelles are responsible for cellular metabolism: chloroplasts and mitochondria. Chloroplast is considered the kitchen of the cell since it is responsible for food manufacturing through the process known as photosynthesis. It converts the light energy it captures to chemical energy in the form of glucose. Chloroplasts can be found in plants and photosynthetic algae. Instead of chloroplast, photosynthetic bacteria have membrane-bound internal structures. Chloroplast has an inner and outer membrane. Within its enclosed space, known as stroma, flattened membranous stacks called thylakoid can be found. Stacks of thylakoids are called grana. Chlorophyll, the molecule that captures light, and other enzymes needed for photosynthesis can be found inside the thylakoids. Mitochondrion or mitochondria in plural form is considered as the powerhouse of the cell. The chemical energy or glucose from photosynthesis is broken down in a series of reactions known as cellular respiration to produce energy in the form of ATP or adenosine triphosphate. Similar to chloroplast, it has an outer membrane that defines the shape and an inner membrane. The inner membrane, however, is arranged in a series of folds known as cristae, where ATP production occurs. The enclosed space is filled with a semi-fluid substance called matrix, where most metabolic steps in cellular respiration occurs. All eukaryotic cells have mitochondria, but large numbers can be found in cells requiring energy, such as muscle cells. Aside from being both energy converters, chloroplasts and mitochondria are involved in the endosymbiotic theory. According to this theory, the mitochondrion and the chloroplast were once free living cells. The mitochondrion was an aerobic prokaryote, while the chloroplast was a photosynthetic bacteria. Both prokaryotes encountered a larger cell, but rather than being digested by or killing the host cell, these prokaryotes survived and together they thrived. The larger host cell provides a comfortable, safe place to live, and the prokaryote, now an organelle, repays the host by making energy that the host cell can use. This happened a long time ago, and over time, the organelle and the host cell have evolved together. Now one could not exist without the other. Today they function as a single organism. During the 1950s and 60s, scientists found that both mitochondria and chloroplasts inside plant cells had their own DNA. It was different from the rest of the plant cell DNA. When scientists looked closer at the genes in the mitochondrial and chloroplast DNA, they found that the genes were more like those from prokaryotes. This tells us that organelles are more closely related to prokaryotes. Another evidence is that mitochondria and chloroplasts also have ribosomes that more closely resemble those in prokaryotic cells than those in eukaryotic cells. Finally, like prokaryotic cells, mitochondria and chloroplasts reproduce by fission, independent from the rest of the cell. Before we continue, let us have a small break. The next set of parts contributes to maintain the structure and shape of the cell. The semi-fluid substance inside the cell is called the cytoplasm. The fluid portion of the cytoplasm is called cytosol. And the cytoplasm is actually 80% water, containing proteins, carbohydrates, lipids, ions, and other low molecular weight compounds. In eukaryotes, organelles can be found spread out in the cytoplasm. The plasma membrane found in all cells encloses the cytoplasm. It acts as the gate of the cell since it regulates the entry and exit of materials into and out of the cell. It consists of a fluid phospholipid bilayer in which embedded proteins form a mosaic or irregular pattern. Similar to prokaryotic cells, integral and peripheral proteins, as well as glycoproteins and glycolipids, are found in the plasma membrane. Unlike prokaryotic cells, the eukaryotic plasma membrane also contains cholesterol for further rigidity. In addition to the plasma membrane, a cell wall can be found in specific cells. The cell wall is responsible for the shape of the cell and protects the membrane and the cell interior from adverse changes in the environment. As mentioned in our previous lecture video, bacterial cell walls are mainly composed of peptidoglycan. The chemical composition of these cells is used to differentiate major types of bacteria and are sites of action of some antibiotics. Not all eukaryotic cells have cell walls. Both algae and plants have cell walls composed of cellulose. Fungi cell walls are made up of carbohydrates such as chitin or glucan. Eukaryotic cells have a complex internal structure consisting of extensive protein fibers known as cytoskeleton. The cytoskeleton provides support and shape to the cell. The three cytoskeletal filaments are, in increasing thickness, microfilaments, intermediate filaments, and microtubules. Aside from size, they also vary in their protein composition. Microfilaments are consisted of thin actin filaments. Aside from maintaining cell shape, they are involved in motility via muscle contraction, cell division, and movement of organelles. Intermediate filaments are combinations of keratin, lamin, and other proteins. Aside from maintaining cell shape, they anchor nucleus and some other organelles. Microtubules found in cilia and flagella consist of alpha and beta tubulin dimers. Aside from maintaining cell shape and are involved in motility, they move chromosomes in cell division, form cell plate during plant cell division, and involve in growth of plant cell walls. How can cells interact with another cell? Since the cells are enclosed within a plasma membrane and sometimes with cell walls, there is a need to communicate with neighboring or adjacent cells. Plasmodesmata are channels or tunnels in the cell wall of connecting plant cells. It allows the conduction of water and nutrients, in some cases spread disease. In animals, junctions are present to connect the cells to another. Tight junctions seal or fuse cells together, forming an impermeable membrane. Adhering or anchoring junctions link the intermediate filaments of similar cells, such as cardiac muscle cells. Desmosomes are even stronger connections that join the intermediate filaments of neighboring cells. Gap junctions, similar to plasmodesmata in plants, are protein channels in the plasma membrane allowing passage or exchange of ions, nutrients, and other small molecules. Some parts are necessary for movement within and outside the cell. Cytoplasmic streaming refers to the movement of the eukaryotic cytoplasm from one part of the cell to another, distributing nutrients throughout the cell. Earlier, we mentioned about microtubules and the cytoskeleton. During cell division, microtubules from the centrosome are responsible for the movement of chromosomes. In animal cells, each centrosome contains two cylindrical structures called centrioles, which also facilitates in cell division. Aside from this type of movement, some cells need to move from one location to another. Cellular locomotion is mainly due to the presence of cilia and flagella. Cilia are characterized by numerous but short microtubules and exhibit oar-like motion. The lining of the nasopharynx and the trachea are covered in cilia. These ciliated epithelial cells remove mucus, bacteria, and other debris from the lungs. Another example is the lining of the fallopian tubes. The cilia here are responsible for helping fertilization by movement of the egg towards the uterus. Flagella, both found in prokaryotes and eukaryotes, are longer but fewer compared to cilia. They are usually anchored to the plasma membrane or cell wall. Flagella move in wave-like manner. An example of eukaryotic flagellate cell is the mammalian sperm cell, which uses its flagellum to propel itself to the female reproductive tract. The last organelle is a vacuole. Eukaryotic cells contain vacuoles or cubicles that act as a storage for nutrients, waste, and poisons. Interestingly, animal cells usually do not contain vacuoles. If animal cells do have vacuoles, they are much smaller than those in plant cells. It is enclosed by a membrane called tonoplast. In plant cells, a central vacuole takes up 90% of the cell volume, mainly for storage of water. All of these organelles, with different functions, work together to enable the cells, whether prokaryotic or eukaryotic cells, to grow, reproduce, and survive different environments.